All right, so it's a great pleasure to introduce today's speaker, Robert Fisher. So Robert is, I'll do the formal bit first and tell stories. Robert's the, if I may, Robert's a research scientist at Usgs in San Diego and also adjunct faculty at San Diego State. And I guess we go back a way. Robert is sort of a, to those who know him, is sort of a force in herpetological systematics and biogeographic studies. Well, very fine work to tell about today on Southern California, but also Pacific reptiles and amphibians. He's done a lot of very important work there. I guess we've known each other since you were a grad student.
Undergrad.
An undergrad, yeah. Good God. But at one point I did have this great passion to work on tropical Pacific islands, because of course it's a beautiful place, why not? So I was working towards this big study on geckos and started to apply for grants to go island hopping across the Pacific and drink pink gins and catch the odd lizard. And I was just about there, and Ted Case contacted me when he was at UC San Diego and said, "Oh, I've just got this huge grant to work all across the Pacific. We'll send the animals to you to do the genetics." So my part of this grant, while Robert and Ted and the others were drinking the pink gins, hopping around the islands, was to go to the quarantine facility at the Brisbane airport and murder animals. Well, they had all the fun, and I've never forgiven it. Nonetheless, we're still collaborating. And it's great to have you here today, Robert. So he's going to be telling us about his work in Southern California. That's the title.
Great. I've removed most of the methods out of my presentation just because I realized it was too long, and I'm famous for going too long on talks, and so I really want to make it on time.
I'm going to cut you off.
Yeah. So what I did do was I brought a handful of reprints that I'll leave at the museum that has the methods to go with the statements I'm going to make. So you just have to accept it, and then you can send what afterwards. You can kind of work your way through them. Two reprints in particular I'm going to point out, which are partnerships with collaborations here with the museum. One is with Sean Scoville, Tate, and Vance Friedenberg on conservation genetics and frogs, and I'll reference, if I have time, some of that work. And then the other one was with some of the folks here, Jim and Michelle and Carol and Ted, on Fragmentation. And so we've been collaborating. I'll use some terms loosely in my talk, we and I and they, and I'll point that out later. I'll define those terms. But anyway, like I said, I've brought a stack of stuff, and and what I've done is I've removed methods and put videos in instead, because I figure it's lunchtime, so we can watch the movies as we go along. First, I'll give you some background. So this is just the outline for the talk. Is what is USGS science? I'm a USGS scientist, research scientist, and I do biological data mostly, biological research. I'll review kind of the Southern California, all the stuff you kind of know, this seminal work that Dave and others have done about why it's a hot spot in Southern California. Review issues about what we're doing for conservation planning in Southern California, how we're applying the science, or what are the issues, and then later about how we're applying the science. And I'll review fragmentation, both direct and indirect effects. Talk about the modeling we're doing, genetics we're doing, natural history, monitoring, both all to understand impacts to then inform how we're making decisions on land use planning. And then I'll talk about fire impacts, which are potentially climate change impacts as confounding factors once you kind of have these great plans and then you just burn everything up. And then kind of where we're going after that. For those that don't know, or those that do know, USGS is the science agency for the Department of Interior, and we're part of the Department of Interior. There's some number of employees that goes up and down depending on who's in Congress and what the projection is for next year. We were in disciplines. Now we're in program areas or mission areas. They've just reorganized the whole thing. We run a bunch of sensor networks for hazards, for water resources in the country, volcanoes, earthquakes, all the stuff you guys know because you're close to Menlo. You probably see people on TV all the time. And in biology, this is the way we were divided up, and we're still kind of divided up that way. About 1,600 employees, 18 science centers, and 40 co-op units. I'm part of the Western Ecological Research Center. My office is here in San Diego. That's where our field station is, right next to the airport, which is really convenient. I can just walk. Except last week when we couldn't fly back from Vegas. Because if you don't know, we had a power outage last week. 5 million people in San Diego and Southern Orange County had no power 2 days before 9/11, and it turns out one guy with a substation in Yuma was doing his job, and the power went down there. The backup generator was San Onofre, which then went off, and then it took a while to get the power back up, which is amazing how fragile our system is. And that ties back to then kind of why we have all these initiatives for solar and wind and other power that we're now pretty heavily involved in. We're the science agency for Department of Interior. We work with these partners, Fish and Wildlife Service, BLM, National Park Service. And we have these different kind of themes that we focus in. I work a lot across themes, doing integrated science with other folks in USGS across our disciplines. We have, in my lab in particular, we do a lot of survey design, database management, data analysis, decision, and then you know, in biology overall, you know, they've been involved heavily in the GAP program, the Breeding Bird Survey, and then the Army has examples of national programs that are run in the former biology of USGS. So what does Southern California need? USGS can provide. Kind of what's our role down there? Why do I kind of have a job down there? And we apply objective science to the landscape in Southern California. As USGS, we're the only federal research organization that is non-jurisdictional and can focus on non-marine systems. And so we have a role there that's really different than everybody else because we can just kind of do anything anywhere. Well, that's how I view it. And thus we can study terrestrial systems regardless of jurisdictional boundaries. We have issues with private lands. We partner with private folks, but with regards to other public lands, we can go anywhere. We're boundless. What's unique about Southern California? Well, you know, it's high biodiversity, diverse habitats, and multiple stressors, and 18 million people. These are just, you know, a lot of diversity, a lot of rare and endangered species that only occur there. A lot of habitats. There is still habitats down there. A lot of people don't believe it, but there's lots of stressors. And as we know, there's continued urban growth. We, you know, again, lead the country in urban growth. We lead the country in federally listed species. And California as a whole is, you know, one of the leaders in the country for extinct species. And so there's a lot of conflicting needs in Southern California. The need to develop and the need to conserve species. So beginning in the 1990s, habitat conservation plans were viewed as a solution to this land use planning. Habitat conservation plans are, if you don't know, it's a piece of the Endangered Species Act that allows the Fish and Wildlife Service to engage in planning on private lands for non-listed species, and it's the only nexus to have control or regulatory control over non-listed species used through habitat conservation plans. Southern California has been touted as a model for this approach. I mean, you guys are, I'm sure, are aware of habitat conservation plans. You have plenty in the area. The model was a few species also could be surrogates for other species and communities. So they're, in California, HCPs are conducted with Fish and Wildlife Service and California Fish and Game. They have the NCCP program at Fish and Game, Natural Communities Conservation Program, too, so they can partner and sign these MOUs with landowners and jurisdictions. Initially, 6,000 square miles of Southern California was the focus starting in 1992. Now it involves about 7 million acres across the state. And it uniquely, like I said earlier, gives Fish and Wildlife Service authority over non-listed species. I'm just going to try to self-regulate myself here. Feedback loop can exist between planning, policy, science, and funding. So our role for the last 15 years has been, you know, part of this feedback loop. We're doing the science, we're in the meetings, talking with the managers, and trying to kind of do informed and adaptive management as we go along this process. So this is done, again, by conducting science, serving as oversight and nexus, and assessing relevance science to these different processes. So that's what I do instead of teaching, is I sit in meetings with lots of different people. I'm involved in different committees at national, state, and local levels. Basically land use planning involves all these things. You know, needs funding, utilize the science, and involves stakeholders. And so we're engaged at all these different levels. This is an initial habitat conservation plan map as they divided up Southern California. And these are the plans like MSCP, Central Coastal Orange County, Western Riverside. A lot of those plans are done. And when we say done, that means that there's a diagram. It's got where you can build, where you can't build. And we consider, I call those biodiversity futures. That's the decision on where the future of biodiversity is going to be. So we then can test those hypotheses as to whether they're correct or not. Are they maintaining biodiversity? are they maintaining ecosystem integrity? And so most of the work we do is focused on those two questions: understanding ecosystem integrity and trying to understand and monitor biodiversity. So this is a recent lab photo of the group that works for me. Some of the folks now work for Amy Vandergast was one of my employees, and we had a team, and then now we're peers, and she's got a couple of folks that used to work for me, but I mean, there was nothing wrong with that. It was great. We elevated her to a PI, like I'm a PI. The terms that are important is, when I say I or me, I mean we. When I say me or us, I mean they. And when I say they, it's like something I heard about somewhere. That somebody's done something. So I just want to be honest about that. Those are important terms to remember as I go through this. Okay, so we have these multiple stressors in Southern California. We have direct impacts, footprint of urbanization, which is what everybody wants to focus on. They don't want to focus on indirect effects because they're much harder to get to. So you can do an XY map and say, here's where we're going to protect, here's what we're not going to protect, and everything we can protect is going to be fine. But we know that fragmentation, contaminants, exotic species, all kinds of stuff are going to happen in those areas. And so this indirect effects is a really key piece of the science we're focusing on and trying to educate people about. Educating people, meaning managers and landowners. So basically, let me step through this real quick. I forgot to delete those. So one of the things we're interested in, and this is really cool. Oh, I think it's really cool. So this is a map of flooded Southern California. So if we go back into the Pleistocene, we flood the landscape. This is what it looks like. So for me, as a biogeographer, I think, well, you've got all these little islands, and you've got really interesting stuff. So if we're going to urbanize Southern California, the terrestrial part, let's urbanize the flooded part first, which is what we've done. Now, what's at risk is the old growth, the old islands that actually weren't flooded and washed clean during the Pleistocene high and low sea level events. So these are the places in the landscape that are still emergent, but now are what we're planning for. And so a lot of this stuff is informed by Craig's work. I mean, like the models of the stuff they did in the wet tropics. I mean, that's how we're trying to think about this landscape. So what we're doing now is planning for the stuff that we shouldn't be building on anyway. But because the stuff that already was the least important potentially for biogeographic processes and has already been urbanized. That all makes sense. So here's so we think about then urbanization and fragmentation in multiple ways so that we can then ask these questions. So here's the road matrix in Southern California. Here's fragmentation of aquatic systems. Basically, these are dams and diversions, so we have very few watersheds that are intact that you can think about as where steelhead can run, where you have headwater streams that are connected to each other via downwater portions. And then we've developed, we published this in 2004, our development impact model, where we've taken then all these pixels and scored them for their kind of how we think they might serve for biodiversity. And then this is then basically a direct impact model, not an indirect impact model, right? So this is then fuzzy edges, 500 meters, saying that may be where species are impacted by Argentine ants or blah, blah, blah process. And so, but now we want to think about indirect effects as well, like nitrogen deposition. We know from literature that, these are they, but actually you've seen it, that the San Bernardino Mountains get the highest nitrogen deposition in the country and so if you are sensitive to nitrogen in the stream habitat, here and the Front Range in Colorado, those are the two places that are the worst place to live in the country. And so, so we know that these indirect effects are really pretty significant. And for this talk, I'm focusing on both Santa Monica's, Orange County, this part of Orange County, and then San Diego MSCP. So I'll show examples from those, but I just want to lay those out. So if we think about indirect effects, think about light pollution. So if you're a heteromyid rodent and you like dark nights, because that's when you're active because of cats and owls and other things, then the best place to be is Camp Pendleton here, B, because this place, LA and San Diego, it's going to be bright all the time, and there's not going to be a lunar cycle that you can see. So it's washed out. So these are the types of indirect effects. This is just light pollution from this atlas, light pollution. Sorry, I'll step back. So LA, San Diego, Camp Hamilton. And what this is saying, so if you're a glossy snake or a long-nosed snake and you are feeding on heteromyid rodents and they're declining because of light pollution, you might be declining because of light pollution as well. And so there's some of these indirect effects where we're seeing declines of species that shouldn't be declining because of the size of the reserve, but they're declining because of indirect effects that we don't yet understand. So a lot of what we started with, I says, how do you even find and count and measure these things? And that was a big piece of what our work was starting in 1995 when I started my postdoc with Ted. And so we started using pitfall traps to look at reptiles, amphibians, small mammals, and invertebrates. And then I've been testing protocols over time. And in particular, like I said, we have a lot of endangered species. Some of those species are naturally rare, naturally cryptic, naturally hard to find. And so we've got to develop protocols that we can robustly use to monitor those things. So here's a Pacific pocket mouse, Camp Hamilton. It's one of the smallest mice in California. And it's, I just want to show you this little video. This is one species we're working with because here's a heteromyid rodent. We think it and glossy snakes still occur at Camp Pendleton. We're trying to understand why they still occur there. And we're trying to figure out how to monitor for this thing. This is our little trap array. We have different sized traps that are set up in different ways. We have these tracking tubes. And if you watch a mouse, this is the kind of stuff that we have to do to understand even how to monitor. So here's a pocket mouse there. And you can watch the behavior. You know, we standardly use small mammal traps. We standardly use lots of different things, but without kind of testing the efficacy of these things in a robust way, we don't really know what we're sampling for and in what conditions. And so we watch the mouse hanging out and feeding, yummy. And then here's another mouse. So far, neither have gone on a trap. Oh, I like you. Let's hang out. Okay. And then you can just see. So we set up these arrays of cameras so we can actually understand their behavior and understand their behavior as they're interacting with these traps that we're using to actually trap the mice. And neither mouse actually went into a trap. And so we can watch, and some of these traps, even the smallest traps, the mouse will go back and forth in and out of the trap 20 times, and it won't go off. And these are super sensitive traps. I mean, so without this kind of data, when we're trying to monitor super extremely rare species, it's really difficult. So now we've got a mouse that's super small. We know it doesn't use the traps really well. So then let's use a bigger tool. Let's take a dog. And so now what we've done is we've set up grids, and we're using dogs a lot more to help try to find stuff. So there's this dog has a search pattern he's going to do, and he's looking for pocket mouse scat, which is a tenth the size of a grain of rice. And so what's actually the volume's on? It might be. So the dog is, and this is then telling us where to even start now looking for mice. So then we take the scat, we genotype the scat, and we can tell which species of mammal it is, whether it's a mouse or whether it's target or non-target. And we're going to be using these dogs for badgers soon too, the next 2 months. So now that this is the tracker. This is from University of Washington. These are conservation canines, and we've contracted with them to come down and do this work. They use the same dogs on ships to find orca, to collect a whale scat, which is amazing. The same dogs. Yeah, they sit on the front of a boat. That way, that way, that way, that way. It's amazing. But here it is. So now he's found scat. So then she's got to come over and check out the scat. And then we look at it. Is it real scat? The dog gets a toy, collect it, and then we can genotype it later. And so I'll show you what the scat looks like. So this dog is going across Camp Pendleton. This dog just went through coastal Orange County looking for pocket mouse sign. Didn't find any there because it used to be real common near UCI, and Dick McMillan used to have grad students that worked on it. So like I said, we're trying all kinds of tools to try to think about how to best look at this. I just got to show you the scat real quick. I tell you videos, how about that? And so this is some of the stuff we're working on writing up right now. And like I said, a lot of the stuff's not in the handouts, but it's in reports we've submitted to partners. What I didn't say was that I spent about $1.5 million a year on salaries, and of that, a small portion is federal money that's kind of we consider base. So that,
he was kind of going around here, and at first he kind of went here, but then when I had him show me, he went here. Not at any of these ones, and I do see a scan right there.
Is that crazy? So then we collect that, and we genotype it. And say, is it a pocket mouse or not? I mean, that works. That's real science we're doing. It's crazy. And that dog's got a GPS on his back, so we know exactly where he went on the landscape and everything. Then we, like I said, set up these grids, and then they work the search pattern. Months of doing this. So then we look at also these, like I said, that edge area that is between what we consider non-suitable habitat for biodiversity and suitable habitat for biodiversity. That focal area is a big piece of the science we do. That's where connectivity might take place. That's where edge effects take place. And so we think about the effects of roads and buildings and parking lots and all, golf courses, and as they affect the function of these reserves and the integrity of these reserves. So in some cases, these are the kinds of roads we have. I think you have the same kinds of roads up here. And what we can do is we can then test different species like Ketodipus or some lizards where we trap them. We have set up traps, and then we dust them with fluorescent dust. And then you can track the animal. And then we can run these models. I didn't show the model. You model where the animal might go, and then you can test their responses to roads against models of that to show that some species, in fact, avoid secondary paved roads and highways. And that's what we need to understand when we're talking about then the genetics and the genetic consequences of fragmentation by roads in these landscapes. And so then we can also radio track animals. We radio track rosy boas, red racers, mountain lions, a whole suite of species. And then we can look at their home range patterns. Like for rosy boas, they don't go anywhere. They kind of hang out year to year to year in the same spot. We know that we've done genetics across rosy boas, and we have an understanding of why they're so genetically diverse is because they don't go anywhere. But they don't cross roads. Even in this landscape, they very infrequently cross dirt roads in this landscape where we have multiple individuals of rosy boas. And that's consistent with what we understand as their historic genetic patterns. So then, this is for turtles, for western pond turtles, which is one of the focal species we spend a lot of time on. We know that nationally, our demography is changing. You're going from having male, female turtle populations equals numbers to now most populations of turtles, or lots of them in urbanized areas, are male skewed. The females go to lay eggs, get run over. Go to lay eggs, get run over. So you have males left. And so in Southern California, we can look at that, and we can see what's the distance to a road and how many males, females we have. And then, and so these are 3 examples of pond turtle populations with the same amount of trapping effort, the capture rates. 6 males, 0, 11, 1, 10, 9. And so now what we're also doing is we're genotyping all these individuals, and we can show that actually each watershed of pond turtles now with our SNPs, so we're partnering with Schaefer and Full Snakes, we can identify unique watersheds. We can identify an animal that was moved to another watershed maybe 10 years ago, and we can still pick up that signature of that individual. And so we're kind of come up with strategies to restore these areas, to fence these areas, to come up with, how many places can we have turtles, and how many, should we be effective at conserving turtles in Southern California, but informed by understanding the landscape matrix as well as the genetics of that landscape. And then here's a really cool thing. I got to show you another. This is really neat. So this is our bobcat, number 10. We got a whole bunch of bobcats in this area. This is northwestern Orange County. Dave will like this. This is Ware Canyon, where we have both Pacificus and or Major and Niagara Ventures together here, and over this is one lake, so down over here. So we've done a lot of work in this area. We tracked lions, we tracked bobcats, we did lots of herp stuff. And so what we're going to do is we're going to make a fake bobcat. And so we can take the data from our transmitters. And this is GPS collar data on bobcats, 15-minute intervals. We're collecting data. We can then create a landscape that we can then run them through. Again, now this is 200-meter edge, and we've modeled that edge. We've tried 50, 100, 200, 400. How is a bobcat interacting with the edge? And 200 is the best fit for the model. Then we can take bobcat data for B10. We've got a paper in press right now, or we've submitted, not in press yet, but on kind of this process. We can look at data. We can look at how they make decisions from point to point, and how they're interacting with that edge, and then we can make these individually based models. And so there's the home range. Here's now 50 fake bobcats making 500 moves. And then we can look at how these fake bobcats match to what the real bobcat does. Because what we want to do is we want to optimize our model as best as we can so that we can then run fake bobcats in other landscapes that we don't have data. So we can learn about how they're doing connectivity. We also have data on the diseases of these bobcats. We have data on the genetics of these bobcats. We've got a paper we're trying to get published right now showing the differences between these patches and bobcat genetic diversity. So now we can take that same, put it in southern Orange County where we don't have bobcat data, and then run another 50 bobcats. And just inform ourselves about that landscape so we can then better learn how they're thinking about it. And then one thing we do learn is that bobcats are left-handed, and that they make left-handed turns, which is really interesting. At least the B10 is left-handed. But it's another way we're thinking about the landscape, tying that back to real data, tying it back to the genetics of the landscape. But you can see, even from this, there are real similar patterns in these different landscapes, and then that tells us something about connectivity through a bobcat's view. And then we're thinking about conservation genetics and landscape genetics, conservation actions at multiple scales. Step through this pretty quickly. This is a paper we published a couple years ago on this, what we're calling multi-species genetic landscapes, where we're looking at genetic divergence, and we can identify areas on the landscape. And this was some stuff that we and Craig and others had discussions about early on, about kind of these genetic landscape analysis. These models aren't informed by species distribution models, and we're thinking about that a bit more. And then we can kind of come up with this overview of what's the divergence on these genetic landscapes overlaid on protected areas. So we can identify kind of hot spots of what we consider genetic divergence that aren't actually protected. So we can then focal attention, and if there's money again, purchasing, protecting lands that are maybe important. And then when we look at conservation fragmentation genetics, this is the Santa Monica Mountains. We've published 2 different papers, one on crickets, one on 3 lizards and a bird, looking at how this fragmentation in this landscape across time has affected their genetics, and can we actually see a loss in genetic diversity, and in a 70-year period in one small landscape across crickets, lizards, and a bird. And we've actually detected that and published that. And so that's really fast that these things can be changing in these landscapes. And it's a cricket. There should be plenty of them. And then we're looking at stickleback. We're going to be looking at Santa suckers. Looking at pontorills. I'll just show data we're just now generating for red-legged frogs. These are the 3 southernmost populations of red-legged frogs. 2 in LA and one in Ventura County, and then these are red-legged frogs through San Bernardino, Santa Barbara, then kind of part of Ventura, and then up through San Luis Obispo. So we can see these southern 3 come out when you use this K of 2. When we change it to 6, we see that each of these 3 populations come out separately. This one is in the wrong spot. It's actually part of Aliso Creek. So this is Ventura, and then 2 different LA populations of red-legged frogs. This population went through a significant bottleneck in a post-burn bottleneck in 2005. So we tested before and after the fire and the bottleneck, and it seems like it was bottleneck before the fire. Even though there was 300 to 500 individuals, they dropped down to 25. Now they're back up to 150. It's really seems like they couldn't lose more genetic diversity because they already had been bottlenecked. Versus the paper we just published with Vance and the team from Berkeley, where we showed that for City Creek, for yellow-legged frogs, they actually, when they got bottlenecked after the fire in 2003, they lost 50% of their genetic diversity. So with 2 different frogs in 2 different sites, both affected by fires, but these guys we think have been bottlenecked previously, and they're still persisting in that landscape. But we're trying to decide how to do restoration in Southern California, and so now we were trying to figure out kind of what's going on. Can we use 1 or 2 or all 3 of these populations? But it looks like we want to think about each of these populations a little bit differently. So then fires have recently changed focus. We're thinking about all these edges, and you have these reserves, and we know how much habitat there is. And then what we did was we ran this experiment. In 2003, we burned up the landscape. And then we replicated that experiment in 2007, burned up the landscape. And so now we can then test the resiliency of these reserves against burning up the landscape. And so a lot of the focus we've done since 2003 has been looking at the effects of these landscape level fires, both in the terrestrial integrity of that landscape and in riparian function. And so the truth is Southern California is a fire-prone landscape. Santa Ana winds and drought create firestorms. The myth is that these fires are natural and this is a natural process, because in reality, the fire starts are not natural, and we don't naturally have fall fires in Southern California. These were human causes, and that we have we are trying to test whether the physical and biological landscape does have or does not have the resiliency for these new pattern of large fall fires. So here's an important fact: the 28 largest Swab fires. It hasn't been updated since we had the station fire, which sits right here, which is from LA when we burned up half the San Gabriel Mountains. And when I say we, I mean they. Burned up half the San. But basically 60% of the last of these fires are from Southern California. 100% of these were human caused, and 58%, which is now I think it's like 67% or whatever, were since 2000. So is this a climate change effect? We don't know, but we do know that it has to change the way we think about this landscape, because we can't just say, okay, we got these green areas, they're fine. We got this edge area we're going to focus on, and then we have the rest of this that we don't worry about. But fires are really changing that thought. Not only are they burning, but they're burning the same places over again in rapid succession. So here's, let's see. Yeah, so there's 60,000 acres in San Diego that we reburned between 2003 and 2007. So we've got all kinds of experiments. Burned once, burned twice, burned once later, burned, and then this is what happens after a fire. So you have City Creek, you've got Nice Creek, waterfall, then you get this after you get the big rain events. So part of the science became, how does this go to this? And what's the mechanism of that? Because we know what happened was this took 100 frogs and reduced them to like 3 when it became this. But what was that mechanism that reduced them? Was it direct and covered in silt? Was it, what part of that? So a lot of our work is focused on understanding this mechanism. So here's a real simple model. You have sediment stored on upslope vegetation because we didn't burn a lot of these slopes for a long time. So you got a lot of sediment. You burn the organic, the rocks fall down. And I'll show you what that looks like. This is in Orange County. This is rocks falling down. Because now these are big rocks. I'm standing here, and these rocks that are falling, you'll see these ones hitting the water. Some of these are, 50, 100, 200-pound rocks. This event knocked out the resident population of trout in this canyon. This is in Harding Canyon in Orange County. And this is post-San Diego fire. I mean, this is a yucca. That's a big plant. And this is just, one day. This is like 3 days after the fire. And and the whole hillside just collapses, and you just watch the pools fill in. These are big rocks. So that's where it's like, once it got really crazy, I had to back off. So we have this, which is called dry Ravel. So this actually starts happening while the fire is going. So to test this event, you actually have to be there, because as soon as it's burning the organic material, the inorganic starts moving down the hillside. So now I've said that the creeks at the bottom are fragmented. So these are headwaters. So once you take out these headwater streams, there's no way to recover because you've got channelization below it. And then we have a second technique. So this is the pool when it's nice and pretty. This is the pool right as the fire was passing. It's covered in ash and such. And then this is that same pool after the first rain event. So this is that dry Ravel slope I was showing you, but now it's eroded into this pool. below it. It filled up the pool above it and then eroded in the pool below it. Yeah, that's that same slope. And then we have this other thing that we call debris flows. So we have two ways we can destroy headwater streams, not just one. This is now after we get a rain, and under certain conditions, you can actually get the landscape to migrate. And so this is the Christmas Day storm, killed 16 people. This one event after the 2003 fires. And this is coming off the San Bernardino Mountains. And here's what, now this is, watch for the mailbox. You'll see a mailbox right here. This is a street and the cars are already gone. They're down the hill. Oh, hang on. There's a mailbox. Look at the size of these boulders. Now this is after the cars have been migrated down the hillside. This is after it came out of a canyon. So this is coming out of a canyon down a city street. So now if you're a newt and you're in that canyon, yeah, it's going to be a little tough. Or a yellow-legged frog. Like I said, I want to show you guys videos. Isn't that cool? But then we can also do this thing. We can burn up trailer parks and critical habitat. We can do all kinds of fancy stuff with these fire experiments. And then we can look at the effects of what happens to this material as it gets into the system. So one of the things that's really interesting is if you sample ash, well, and now this is work I'm doing with these geophysicists and geochemists in Denver and also USGS. These are the guys that worked on the 9/11, the World Trade Center dusts. And what they found is that if we take residential wildfire ash and we just look at the pH of leachate in that wildfire ash, here's where it sits. It's like a 13. And if we compare that to common things like milk and magnesia, we compare it to liquid drain cleaner. This is what is now right next to the creek that's going to flush in. So we have both geologic and chemical ways we can destroy biodiversity after these fires. And then this is burnt soil, and we've got a lot more data now, but it's pretty bad after these fires. And there's lots of ways you can make it bad. And so, like I said, we're trying to understand how do we create resiliency in this landscape? How do we make more populations and more species so that we have resiliency for this? This is a list, just a tally of decline and extirpation events of different TNE species in Southern California. We've documented since 2000. There's more than that. These are just ones we've documented that we've been monitoring. So we're losing stuff really fast, even endangered species, even in protected areas. So here's the yellow-legged frog. Here's our captive breeding facility for yellow-legged frogs. And we're trying now to experimentally reintroduce these into San Jacinto Mountains. And we know the genetics of this population, because that's in our paper as well. And we know that we have half the genetic diversity that's in the wild, so we are informing ourselves and starting to collect more tadpoles to put back into our captive population. And so, so one of the goals, the long-term goal, is to develop these realistic biodiversity futures. Where is there going to be biodiversity in the future, and what is going to be the resiliency of that landscape? And so that's, like I said, kind of what we're working towards with all the different kinds of science that we're doing in my lab and in partnership with tons of other people. And then the next steps are informing our landscape genetics with species distribution models, linking climate to our species distribution models, running these IBMs, these individually based models like that Bobcat model, running those in future landscapes, using our movement data for rosy boas and for red racers to make models of how they would view these landscapes, and just testing these landscapes in different ways. And then reintroduction experiments informed by genetics, and then continuing our protocol development. How do we find these species we can't find? And how can we find them robustly and repeatably so that we can actually do something towards recovery and understanding of biology? And then one of the big next steps that we're doing in our group, my group and Amy's group, is where money's coming from in the future is the solar development under the desert. And so this is our first assessment of kind of landscape genetics using lots of people's data sets. Jimmy's Cratophytus is in here. There's lots of stuff to inform where are areas that we consider hotspots and how do those then interact with areas that are future planning areas for that. And now this is actually informed by species distribution models. So we created species distribution models for the taxa, then did the landscape genetics for that taxa, and then integrated across all taxa. And that's different than what we did in our 2008 paper, where we hadn't yet informed those models with species distribution models. And so this is kind of our next step in thinking through this, and some of these things just jump out. I don't want to focus on it too much. I want to have it set for questions, but that was what I had, you guys. That was it.
Nice. Robert,
yeah.
Breathe.
Yeah.
I can only admire your enthusiasm and continuing humour in the face of all this. Anyway. So, there's plenty of time for questions and comments and what have you. Yeah.
So the bobcat thing is really cool. What I'm wondering is how many of those bobcats are you looking at? Because individuals probably have idiosyncrasies.
Oh no, we're going to integrate across. That was just based on one. Yeah. So we have 3 of this paper we've just submitted where we're just talking about the technique of how we got to that, and then we're going to integrate across a bunch of bobcats to do that, and same with the lions, integrate across a bunch of lion data to really figure out how they make the decision. Like I said, that one bobcat I think is left-handed, and that's why you see these weird patterns. And then what we didn't do was we didn't make the landscape, so we know exactly what size culverts bobcats use and what they don't use, and so we haven't informed those roads yet with the culvert size and then future culvert size based on build-out scenarios, so that we can run the animals through the landscape now and then through a future landscape, and then say here's where we're going to lose kind of connectivity out there. So we've got several kind of next steps, but that was exciting to actually play around with that now.
I had the same question.
Oh, okay. Sorry. Yeah, that's our hope. A lot of this, like, I just had people email me this, email me. I just tell them I need a slide of this. Yeah, so I just wanted to add a bunch of really cool stuff in there that's new.
So I'm very interested in your comments about fires, and because there is a mythology out there that fires are natural part of the natural landscape, but these fires are orders of magnitude different than natural fires.
Yeah, and so there's this classic argument between John Keeley and Rich Minich. Rich is at UC Riverside. Keeley used to be at Upside Down College. He's now at USGS. About what is natural fires. And Rich says Baja California has natural fires, small patchy fires that are lightning strikes in the summer. And that makes logical sense. John says that's not. He says no, these big wind-driven fires were the natural. And you look at the fossil data, which would be deposits of sediment in the bight here, you can see how they've got calculated how big fires were historically. You can go back through time and look at fires from the 1800s, 1700s, back through time, and get an idea of how often these big events might have been. But the problem is that whether it's small or big, there's too many, and they're too fast. And so, because I've given this talk with mythology, and John just says, "You're wrong. These are natural events." I go, "How can you have a lightning strike in July and a fire in October?" He says, "Well, because the ashes are just still smoldering underground." And it could be true. It could be true. But you're not going to have that many that often. And that's the climate change potential effect from the changing El Nino, where we're getting really, really wet years and then followed by really, really dry years, driving into that. But it's definitely a dogma. And when I said, this isn't my data. This is CDF. This is the size of fire in California. I didn't create that. And you can see who caused it. It's power lines, or people, or arson. And so that's not made up data. That's what it says. It says that they're all happening really fast and in the same place. Yeah. So it's really concerning because that's something we can't control. Right. And so it's really trying to build this.
And it does really wipe out the riparian situation.
Yeah, it does. So one thing we're doing is we're modeling what are the places on the landscape that have never burned, the old growth riparian sites, because then we want to do is create a map that we can give managers. So if there's a fire in this area, this is a resource you have to protect. It's not a cultural resource, it's a biological resource, because we don't want that piece of the watershed to burn, because it's never burned, and we know as soon as it burns, it's going to fill in with sediments. So that's where you might, over time, do chipping and doing some other things to try to reduce fuel loads in some of those areas, just so we can protect some headwater sites for longer term viability.
So you mentioned that you have these biodiversity futures, these maps. How's that actually translated into lack of development? And is that working?
Yeah. So in San Diego, in the MSCP, they're creating the biggest urban reserve, right? They're creating a national wildlife refuge that's 80,000 acres in coastal San Diego County.
What's
MSCP? Multiple Species Conservation Plan, and that's the county and city of San Diego, and it's a southern San Diego kind of plan. And that reserve is 160,000 acres total, but 80,000 of it is actually Fish and Wildlife Service federal land that they're purchasing to put into reserve from willing sellers and stuff. But that's
Where is that? Is
That's
that
Proctor Valley. Proctor
Yes.
Valley. It's the top of Otay Mountain. You know, from so Otay is BLM, and then the north of that all the way up to Sweetwater Reservoir. All that whole area is part of that landscape. Problem is, it's got all kinds of fragmentation issues there, but in some cases it's a blob map, and then as build out occurs, they then make decisions about where to site those houses versus whereas in other places like in Orange County, the Irvine Ranch, you know, made a map. They made a map with the county and with the agencies and said, here's the reserve, here's the 37,000 acres. That's it, you know, here's where we're going to put it. And so that's Limestone Canyon, that's Weird Canyon, that's these other sites. So those are real experiments you can test, because those are really on the ground. You know exactly where those boundaries are, and then you can try to do adaptive management. Now we know that this road has to change a little bit because it's fragmenting this stuff. So, yeah. All right.
So just related, going back to the fires, given you've got such excellent data now tracking what burned when, do you, for any taxa, do you notice just a general decline, or is it sort of like a, you know, it reaches a ratchet point where, you know, 2 fires and it's all gone, or they can survive one? And
no, so we have this paper that came out on some of our early work on reptiles and fires, and then I was going to show some graphs. I wanted to show the shrew graph. It's really striking. So for one side, it's the UC Reserve. It's Elliott Chaparral Reserve, and that hadn't burned since 1945. Burned in 2003. Hasn't burned again. That shrews went there from, you know, we get like 30 shrews a year to 0, and we've been tracking them, and that burned in 03. So we started trapping again in 05, 6, 7, 8, 9, 10, 11. That many years, no shrews. There's been like 3 captures. And Batrachoceps, that many years, 2. And Batrachoceps, we caught plenty there. Dave's got them in his genetic analysis. So what's happening with these one event fires that are so big is they're xerifying the landscape. They're changing the soil, and and this we haven't been able to figure out how to monitor. I've talked to people, and we're trying to figure out how to do it, because you want to put all these sensors out there, but you want to put them so where it's going to burn in the future, but we don't really know. But you can see the soil changes, and so small snakes, the salamanders, the Insatina on Otay Mountain, they're gone. There's a bunch of things, and shrews. Shrews is one of the mammals that disappears. They're not. We're not detecting them. We're not saying why they're gone, but we're saying that.
So do you think that's like a fuel load problem, that the fire is so hot? I mean, could you look at other areas where you've had a fire and then see whether they come back? I mean, is it just an intensity effect?
I don't think so. I mean, this is across multiple stations in these. I mean, so in riparian or wooded areas, in oak woodlands and stuff, we don't see that same.